A second way to validate specificity of your antibody is to use multiple independent antibodies that recognize non-overlapping epitopes. One example would be antibodies targeting either the amino or carboxy terminus of the same antigen. Use a minimum of 2 antibodies raised in either the same host or different hosts, mouse and rabbit for example, to detect samples on separate blots that are known to express different levels of the target protein. In either case, the results should show an identical number of correctly sized bands on the blots. Independent antibodies derived from different hosts can be tested on the same blot using multiplex fluorescent detection with different antibodies of different spectral fluorescence. Co-localization of bands of the correct size in both channels is proof of specificity. A different way to use the multi-antibody approach is to modify the target protein with an affinity tag such as His6, MYC, HA, or FLAG, or fluorescent proteins such as GFP, RFP, mCherry, etc. Then use the modified protein as the target for a well-characterized anti-tag antibody and the target-specific antibodies under evaluation. Corresponding bands of the correct size on both blots are indicative of antibody specificity. It is important to keep in mind that cloning of the tagged protein may affect conformation, trafficking, or function of the original protein. Placing the tag at the carboxy or amino terminus should mitigate this problem. It is also possible that the junction of the original protein and the tag can lead to a creation of a neoantigen that is unrecognizable by the antibody.